The process of understanding how bacteria specifically communicate with the brain it's not very well understood now because of how hard it is to access the tissue. When you have invasion of your gut microbiome or infection with pathogens that can cause things like leaky gut syndrome or IBS or IBD, those diseases can co-occur with neural diseases. So what we're interested in doing is making devices that can detect the presence of a biomarker right in the tissue of where it originated from and where the disease is occurring. And we're trying to find ways to do that non-invasively. What we are trying to do is make a platform where we can have a sort of model gut epithelium, which will include also a mucus layer, and then colonize that mucus with different types of gut bacteria. The most important part of this system is that we are going to incorporate electrodes that can do like in situ sensing of how the cells are behaving at any given time. We want to be able to correlate that to a key molecule that we think is the communication factor between the gut and the brain, which is serotonin. By measuring serotonin and perhaps other biomarkers as well, directly from the gut tissue where it's generated, we hope to arrive at a much more precise understanding of the relationship between gut health and mental disorders such as depression, anxiety, autism, and a whole host of other mental disorders that can co-occur with gastro disorders. And that will help us much better understand the gut microbiome-brain-axis, which is the underlying factor for all these physiological states. Potentially, this sort of knowledge will be able to be used by a physician in the future who can measure the serotonin generated specifically in your gut microbiome and prescribe a treatment just for you. And that potential is very exciting.